Bismillah ar-Rahman ar-Rahim. Prion proteins are believed to cause a neurodegenerative disease by acting as a template to misfold other cellular proteins that cannot be degraded. The word prion stands for proteinaceous infectious agent. The prion diseases may be acquired either through infection, for example, Creutzfeldt-Jakob disease, or these may be inherited by mutations, for example, familial CJD. Although infectious prion diseases represent less than 1% of human cases, they are linked to mad cow disease in the United Kingdom or to cadaveric human growth hormone use in the United States and France, or to the ritual of cannibalism in the New Guinea, where it is called kuru, have received much publicity. So here are a couple of clinical scenarios for prion disease presentation. In this scenario, Q2 diagnosis is cannibalism and prion protein in the CSF. Here the Q2 diagnosis is cadaveric growth hormone administration. This case study depicts the progression of symptoms in prion disease or spongiform encephalopathies. Familial prion diseases are caused by point mutations in the gene encoding the prion protein. The point mutations are changes in one base in the DNA nucleotide sequence. And these diseases have various names related to the different mutations and to the clinical syndrome. Familial CJD arises from an inherited mutation and has an autosomal dominant pedigree. It typically presents in the first decade of life. The mutation lowers the energy required for the protein to fold into the infectious prion protein conformation. Thus, the conversion occurs more readily. It is estimated that the rate of generating prion disease by refolding of the normal prion protein in the cell to the abnormal shape is about 3000 to 4000 years. Lowering the activation energy for refolding by mutation presumably decreases this time to observed 30 to 40 year prodromal period. So that is why the symptoms appear in the usually in the fourth decade of life. When one prion protein becomes misfolded, then it acts as a template through domino effect and causes the cascade of reactions or a chain reaction uh, of changing all the normal proteins into the prion infectious form. The prion protein is normally found in the brain and is encoded by a gene that is a normal component of the human genome. The disease-causing form of the prion protein has the same amino acid composition but is folded into a different conformation that aggregates into multimeric protein complexes resistant to proteolytic degradation. The normal conformation of the prion protein has been designated as PRP-C and the disease-causing form as the PRP-SC. SC for the prion disease known as scrapie in sheep. Although PRP-SC and PRP-C have the same amino acid composition, the PRP-SC conformer is substantially enriched in beta sheet structure compared with the normal PRP-C conformer, which has little or no beta sheet structure and is approximately 40% alpha helical. This difference favors the aggregation of PRP-SC into multimeric complexes. These two conformations presumably have similar energy levels. Fortunately, the spontaneous refolding of PRP proteins into PRP-SC conformation is prevented by a very large, enormous activation energy barrier that makes this conversion extremely slow. Thus, very few molecules of PRP-SC are normally formed during a lifetime. The infectious disease occurs with the ingestion of infectious prion protein dimers in which the prion protein is already folded into high beta structure. These infectious prion proteins are thought to act as a template to lower the activation energy barrier for the conformational change, causing native proteins to refold into infectious conformation much more rapidly. Much and this role is like the role of chaperonins, which assist in the chaperone proteins, which assist in the which act as template and assist in protein folding by lowering the activation energy. The refolding initiates a cascade as each new infectious prion protein formed acts as a template for the refolding of other molecules. As the number of infectious prion protein molecules increases in the cell, they aggregate into multimeric clumps which are resistant to proteolytic digestion. Once an aggregate begins to form, the concentration of free uh, infectious prion protein decreases, thereby shifting the equilibrium between the non-infectious prion protein and the infectious prion protein to produce more infectious prion protein. This leads to further aggregate formation through the shift in the equilibrium, and this initiates a domino effect.